Hello students. In today's session, I'll explain how the cell-mediated and humoral immunity will work in human body. We are learning the topic human health and disease. In this, we have completed common communicable diseases which are caused by virus, bacteria, fungi, protozoans and the worms. To show defensive mechanism for invading pathogens for which our body will treat it as an antigen. Now there are two types of antigen. Antigens are nothing but the chemical components which are known to our body and they are harmless. So body will not have any defensive role against such known antigens. But there are some chemical components, for example, viral capsid which is made up of protein, bacterial capsule which is made up of peptidoglycan, then fungal chitinous cell wall. These all components are unknown to our body. They are new chemical components entering the body. Against this, the body will generate defensive mechanism. So for unknown antigens, the body will generate the defense, and that defense mechanism we call it as immunity. Now in broad way, we define immunity as resistance to disease. It is an ability of a person to fight against all those invading components which are dangerous to the human body. Now the immunity which we have right from the birth, having general effect towards all the invading pathogens, and it is common in all the individuals. We call it as innate, non-specific immunity. Now this innate immunity is working under 4 levels as per the NCERT book. First is the physical barriers. It includes skin and the mucous membrane. Second, physiological barriers, which includes different chemical secretions which are already discussed in previous session. Third is cellular barriers. There are 2 types of cells, phagocytes known for direct attack by phagocytosis and killers derived from lymphocytes. They show indirect attack. Fourth is cytokine barrier. This is specifically by virus infected cells by releasing interferons. These are antiviral chemicals which will block the sites of protein synthesis in the neighboring cells so that viral infection is prevented. So there are 4 levels working in our body will generate the general defense against all the invading pathogens. But there are some pathogens which are directly entering. They are crossing the physiological barriers. Sometimes they are killed by the cellular barriers or cytokine barriers. The left out components after the killing of the pathogens, it is an indication for our lymphoid system to generate the specific defense. For example, so and so enemy entered, we have killed, but be aware. Similar enemy may enter. This is a signal given by this cellular or cytokine barrier. So, lymphoid system in our body will try to generate the acquired immunity towards that pathogen. Now, sometimes the invaded pathogens start giving challenge to our lymphoid system, which is also called as immune system. So, this lymphoid system accepts the challenge and ready for generating acquired immunity. Now, acquired immunity which develops during our lifespan depending upon the type of pathogen or antigen entering the body is said to be specific immunity. This is the most strongest compared to the first line immunity generated by physical and physiological barriers and second line immunity generated by cellular and cytokine barriers. So this we call it as third line defense, which is generated by trained cells. Okay, which I already discussed how the cells are trained and in the thymus and in the bone marrow, and later we call them as T lymphocytes and B lymphocytes. So here acquired immunity which is specifically generated by the 2 special lymphocytes. They get differentiated in primary lymphoid organs and then they're deposited in secondary lymphoid organs, and one group known for generating cell mediated immunity and second generated for humoral immunity. Now cell mediated immunity is generated at the tissue level. The entry of the antigen or a pathogen recognized by T lymphocytes. The T lymphocyte, after recognition, it binds to that antigen and generates the clones. Clones are a group of similar cells which have ability to recognize those pathogens or antigens. I am using both the words, antigen, pathogen. It is one and the same. Now after cloning, there are two groups are made. One group is immediately start fighting with the available antigens at the tissue level, and another group will store that memory, that type of antigen entered, how it entered, and in future it gets ready. Means the first encounter of the antigens is known as primary immune response generated by lymphoblast. They are available in three categories: helpers, killers, and suppressors. Helpers start sending the information through chemical secretion lymphokinase towards B lymphocytes, which are traveling in the body fluids. Meanwhile, killers start attacking on all those antigens and start destroying them, and suppressors are observing. So they are abbreviated as TH, TK and TS. So whatever the role they played in presence of antigen, they gave response to it, is known as cell mediated immunity, and remaining are stored as memory cells. Now after getting information from helper T cells, B lymphocytes who are traveling in the body fluids, they get ready. Now they are not directly relying on the information. They have to check, is the really antigen is available and entering and multiplying in the body fluids and the tissue or not. So after confirming the presence of antigen, B lymphocytes binds to them and they produce their own clones. Now here also two groups are ready. One group is ready to fight, means these are the real soldiers in the battlefield now. Battlefield is a body fluid which is continuously circulated throughout the body. And the second group is a reserve force that we call memory B cells. Now what these plasma cells will do? These, I already told you, these are the real soldiers now ready on the battlefield. Now these plasma cells having the weapons, and through weapons they have to shoot the bullets. So whatever the secretions, I am giving examples as weapons, bullets, just to remember all the terms and the mechanism. So plasma cells are in the body fluids, they start secreting a specific chemical, and that chemical is polypeptide, and this polypeptide known for immunity is known as immunoglobulin, and generally we call it as antibody. Against antigen, antibody is generated. Now what this antibody is? It is nothing but H2L2 composed of 4 polypeptides. H2 stands for 2 heavy chains which are held together by disulfide linkage and arranged like a Y fork. It is a cartoon form of antibody. On either side of the fork, outside, there are 2 light chains, and at the terminal end of this Y fork, there is a specific determination site. It is known as paratope or antigen binding site. The receptors available on the antigen, these are not actually receptors, these are the components available on the antigen are called epitopes. So receptors found on the antigens are called epitopes, and they are recognized by paratopes of antibody. So antigen is an enemy and antibody, okay, is a real fighter. Now this antibody is having two specific regions in each chain, either it is heavy or light. Whatever region I have shown with the pink color, it is a constant region, and wherever I change the colors, it is a variable region. So heavy chain and light chains, which are arranged in the form of Y, having antigen determinant site linked by disulfide bonds, and they get ready to attack the antigen. During our lifespan, we generate two types of antibodies against the specific pathogen, IgE. It is specially during allergic reactions. This I'll explain in the next session, and the most strongest is IgM. Mother's gift through placental circulation and through the milk secreted by the mammary glands on which the infant is nourished, IgA and IgG. So total 4 types. One more is there, IgD. So Ig stands for immunoglobulin, depending upon the linkage of amino acids forming the polypeptide and their ability to fight against antigens. These are 4 types. So when the humoral immunity begins, antigen, it is unknown component entered in our body. It has a receptor site known as epitope. It is recognized by immunoglobulin called as antibody, and it has antigen determinant site called as paratope. So epitope and paratope start reacting with each other. So during humoral immunity, AG means antigen, AB means antibody, complex will form, and the chemical fighting begins. Antigen has to be destroyed either by neutralization or by detoxification or by agglutination, means clotting, or by complement fixing. So these are results of the destruction of antigen. If the antigen is very less harmful, they just neutralize. Antibody is neutralizing the antigen and it will throw in front of the phagocytes for killing. If it is toxic to the body, antibody will detoxify the antigens. The antigens are more aggressive, so antibody will form a clot, and this clot is digested by the phagocytes or destroyed by the killers. Complement fixing. Okay, in Bollywood movie it is a common scene. Only one hero will be fighting and villains are continuously attacking. It is like that. Okay, so a group of villains coming, these are antigens, and only one hero is fighting, that is antibody. So the result of Bollywood movie, it is a climax of Indian movie that hero always wins, and this hero will destroy the multiple antigens. And remember, when we won this fight, the body is destroyed antigens and antibody will remain forever. That's why whenever any infection is generated, don't take immediately medicines. Have faith in the immune system, what we have. Give enough time for that immune system to study the antigen entered. Allow that B and T lymphocytes to generate their defense force. And once they generate the antibodies, we are totally safe till we die. This is how the acquired immunity works. With this, I'll conclude today's session. Thank you.